First of all, I would like to thank the organizers for giving me the opportunity to present my research. Today, I will present to you a new mode of splicing regulation where transcription factor may moonlight as splicing regulators of duplicated ribosomal genes, and I'm going to show you that this regulation is associated with a cellular process, meiosis. The classical view of ribosome biogenesis is depicted as the global regulatory circuit, where both rRNA and ribosomal protein genes are interregulated by promoters and transcription factors, and the increase or shutdown is dependent of growth conditions. However, this process is not really homogeneous. Most ribosomal protein genes are not transcribed to the same level, do not share the same regulatory sequence, and many dRPGs are produced from two genes. A gene-specific regulation, here in green, illustrates that the circuit allows to adjust the amount of ribosomal protein of each gene pair by a post-transcriptional regulation. This specific regulation establishes an expression hierarchy in which one of the duplicated genes is the major copy. My laboratory discovered that dRPGs in budding yeast are regulated by a different in splicing. However, we don't know how these duplicated genes producing a quasi-identical proteins communicate and why the cell favors the splicing of one. In order to study how the gene-specific regulatory circuit works, we are using the small subunit protein S9 genes as a model. In yeast, RPS9B is the major copy producing the most of protein in the cell, and RPS9A is the minor copy producing a minority of proteins. My published work established this model in which splicing determines the expression hierarchy of S9A and B genes. RPS9A possesses an intronic structure which inhibits its splicing and reducing its expression, whereas RPS9B splicing is efficient. S9B protein can bind to the intron of RPS9A, reducing its expression. When RPS9B is deleted, the splicing of RPS9A is increased, but not as efficient as B. So, the binding of RPS9B to the A intron is not the only factor repressing RPS9A. We suggest that the repression of RPS9A is condition-dependent, and we propose that factors other than B may contribute to the splicing repression of RPS9A. Accordingly, we decide to search for factors that bind to the RPS9A intron and inhibit its splicing in a condition-dependent manner. To identify factors that bind the RPS9A intron and regulate the splicing in a paradox-dependent manner, the experimental strategy is the following. We tag the intron of RPS9A with a small heptamer called MANGO. MANGO tag allows to purify S9A pre-mRNA and protein complexes. To identify the proteins that bind the A intron, we use mass spectrometry analysis of the coprecipitate proteins. Then we selected all the nuclear proteins and we analyzed the effect of their deletion on the splicing of RPS9 using a splicing reporter. The splicing reporter here contains the YFP gene that carry either RPS9A or B introns. A total of 150 non-essential nuclear proteins were used in the splicing assay. As shown in this map, we identified 12 factors that differentially induce the splicing of RPS9A relative to B. Surprisingly, gene ontology revealed that the majority of factors are transcription factors and many are associated with meiosis. This result suggests that RPS9A splicing may be regulated co-transcriptionally in a meiosis-dependent manner. To verify the screen result and determine the effect of this factor on mRNA level, we measure the splicing efficiency and the mRNA expression of the native S9A and B. It's not all mutants that affect the splicing on the chromosomal genes. suggesting that the intron context is important. As you can see here, the deletion of the general transcription factor TAF14 and the meiosis transcription repressor RIM101 have the most impact. Both increase the splicing efficiency and the expression of RPS9A and not B. We have also seen that the deletion of these two factors increase the amount of RPS9A protein, as you can see here on the right. We also found that TAF14 and RIM101 bind co-transcriptionally to the intron of RPS9A by RNA pull-down and chip analysis. Since RIM101 is known to regulate gene expression during meiosis, we decided to see if S9 is regulated in a meiosis-dependent manner. Meiosis is a specialized cell division program producing gametes in human and spore in yeast. During this event, yeast cells adopt a particular morphology, a tetrad composed of 4 haploid spores. This process is also called sporulation in yeast. The graph shows the expression of RPS9A and B at different sporulation times. As you can see, there is an increase of the expression of RPS9A and not B. Then, we analyze the effect of deleting RIM101 on the expression of RPS9A during sporulation. As you can see, the deletion of RIM101 results is a constitutive upregulation of RPS9A mRNA without affecting expression of RPS9B. To ensure that this effect is mediated by the intron, we deleted in the intron and the deletion of RPS9A intron completely abolished the sporulation-dependent induction of RPS9A, and this increase is maintained regardless of the cell state. Now, we have to ask the big question: does this change of RPS9A expression have an impact on meiosis? To answer this question, we have deleted RPS9A and B gene and compared their effect on sporulation. As you can see on the graph, the deletion of RPS9A and not B significantly reduces the number of sporulated cells. It's shown that the minor paralog of S9, which is not required for growth under normal conditions, becomes important for sporulation. We have also observed that this effect is associated to a measured dysregulation of the gene expression of meiotic markers. Looking at the cell, we see in the wild-type strain that the expression of the spore whorl marker SPS2GFP is only in a sporulated cell, whereas cells from the RPS9A mutant also express GFP in a cell that do not sporulate. Chalcofluoro staining indicates an aberrant spore whorl. As you can see in the blue staining, unlike to the wild type, in the mutant, we see that the sporulated cell has a higher intensity of labeling. Together, my results suggest a new model of dRPG regulation, where the expression of the paralog is determined by the multi-factor regulation of splicing. Under normal vegetative growth, the splicing of RPS9A is repressed by the binding of RIM101 and TAF14 to the intron. The splicing of RPS9A is also inhibited by the binding of RPS9B, which help control the total amount of S9 produced in the cell. During meiosis, the splicing of RPS9A increases, suggesting that the binding of RIM101 is destructive to support efficient sporulation. The binding of TAF14 may also alter the paralog ratio, but we don't know yet if it is occur in a specific condition. The message you should take home with you is transcription factor may moonlight as splicing regulator. Regulation of dRPGs may take different levels and do not only respond to change in ribosomal protein dose. And RPGs could be regulated by splicing dependent on the condition. At the end, I would like to thank Sheriff for having accepted me in his laboratory. I would like also acknowledge all the members of the lab. Last but not the least, I would like to acknowledge your funding from the CRSNG. Thank you for your attention.